Hello everyone, welcome to a brand new video. On this video we're going to discuss anaerobic metabolism, focusing on alcohol and lactic acid fermentation. What we want to do is look at the difference between what aerobic versus anaerobic metabolism means. Then we're going to talk about glycolysis and how it's a starting point for a kind of anaerobic metabolism. Then we'll discuss how we can do alcohol fermentation with glucose and lactic acid fermentation with glucose. So let's really get started and look at the word aerobic versus anaerobic. Now, as we look at these words and you're dealing with things like aerobic or anaerobic, we're going to really start off with on the positive end and talk about the word aerobic. And my goal here really should be that we're trying to understand that if something is aerobic or anaerobic, the "an" means the negative. So the word we're going to try to think of is aerobic with aerobic and anaerobic. And so what we're just doing is we're adding a prefix here that is referencing it is the negative. Aerobic means that it occurs with oxygen, that O2 is utilized in this process. Now, a big thing is you're going to make around 34 ATPs extra, so that 36 to 38 that you're going to make of ATPs extra. Anaerobic processes usually only make 2 ATPs, unless we're talking about anaerobic respiration, where we occur without oxygen, but we basically use other things to produce chemical energy, and that can vary dramatically. But for the purposes of this video, we're only going to be making 2. We're not really getting an anaerobic respiration in and of itself. So I just wanted to make sure we understood that. Now, as we get into that, glucose, if you guys aren't familiar with aerobic metabolism and basically how aerobic metabolism works in general, I get to writing and I'm trying to think and write at the same time. Aerobic metabolism basically can be summed up with, if I take a C6H12O6, now we know this could be glucose, it could be other things, but it could be glucose, and 6 oxygen molecules. In the presence of that, we can make 6 carbon dioxides, 6 waters, and about 36 to 38 molecules of ATP. Now, anaerobic metabolism, and if you want to make sure that we understand glucose is the start of that, well, when glucose comes in, glucose, which is outside in the ECF, will then be brought into a cell, into the cytosol, and in the cytosol process called glycolysis, sorry, got to writing a little bit wrong, glycolysis, and then glycolysis can go on to complete cellular respiration. It can go on to complete an aerobic metabolism. Now, by itself, glycolysis does not need oxygen. It is an anaerobic process. It's a standalone mechanism, so it does not need that. So fermentation, because that glycolysis is kind of like a replicative fork in the road. I always say it's not a metabolic fork in the road here, and it's like a metabolic fork in the road. If you've got glucose and you have oxygen, you'll do aerobic processes. And if you don't have O2, low O2 or no O2, you'll ferment. And this is why I was saying that glycolysis is the common thread. It is the metabolic fork in the road when it comes to anaerobic versus aerobic metabolism. They both share it. Now, we're going to start off, though, looking at how do we do, in general, the fermentation of glucose. Now, glucose, remember, C6H12O6, it is a 6-carbon compound called glucose. And glucose, so what I'm going to do is draw just carbon skeleton. 1, 2, 3, 4, 5, 6. Each dot represents a carbon, 6-carbon glucose. And this 6-carbon glucose is going to undergo the process called glycolysis, which simply means sugar splitting. Now, to do that, one of the things we're going to do is we're going to take 2 molecules of adenosine diphosphate, ADP, and we're going to bring them in, and we are going to produce with that 2 molecules of ATP during that glycolysis. Now, I will do videos later where I go in detail about this, but right now, this is what's happening, okay? Now, the 2 ATPs that are produced from that is it. That's all you're going to get. But also what's interesting is you're going to take 2 molecules, 2 electron carriers called NAD pluses, nicotinamide adenine dinucleotide, and you're going to take 2 of these as well and turn them into 2 NADH molecules. Now, we're going to do this in more detail later on in some other videos, but I just want to go ahead and do this one now. At the end of this process, what you're making is 2 3-carbon compounds here called pyruvate. And pyruvate being 2 3-carbon compounds, so let's draw those. So 2 3-carbon compounds. Now, what's going to happen to these 2 3-carbon pyruvates is there will be an enzyme. I'm not going to get into that in this video. I may do a more in-depth biochemistry video, but suffice to say there's a decarboxylase that comes in, and that enzyme will pull off CO2. And this will lead with losing a carbon, and this produces 2 2-carbon compounds called acetaldehyde. And there are 2 of these acetaldehydes produced, 2 acetaldehydes. Now, the CO2 is given off. This is why that this process here of alcohol fermentation is purely not reversible. You can't, that carbon is gone. You can't put this guy back and turn him back in. It's lost. Now, what we're going to then do is take these acetaldehydes, these 2 2-carbon fragments, and what we're going to do that's very interesting is we're going to take our NADHs and we're going to come in and we're going to force the electrons they have into that. So we're going to do a reduction reaction. This is reduction, by the way, and we reduce it, and what we're going to make is, again, 2 2-carbon fragments called ethanol. So ethanol, so I'm going to make a 2-carbon fragment here. We're going to put OH on it, and then another one here. And let's put an OH on it as well. Okay, so now we've made our ethanol byproduct here. Now, there are a variety of species that do this. Basically, the main thing that does this is yeasts, specifically Saccharomyces cerevisiae, and there are some bacteria that are also known to do this. Yeasts and bacteria do ethanol fermentation. Now, I know this is a little bit more simplified. I'm not going into the decarboxylases, the dehydrogenases. I will do videos where I go into the more in-depth biochemistry, but right now this is enough that should get most people through something like a general biology course. Now what we want to do is turn our attention to look at lactic acid fermentation. So at lactic acid fermentation, we're going to again start with a 6-carbon glucose. And with 6 carbon glucoses, 1, 2, 3, 4, 5, 6, again drawing the carbon chains, we are then going to undergo a process called glycolysis, sugar splitting glycolysis, where we're going to take 2 molecules of ADP and we're going to produce 2 ATPs. Okay? Now with that, you are now producing 2 3-carbon compounds called pyruvate. Again, the pyruvate is made. So now we got 1, 2, 3, 1, 2, 3 pyruvates. Now, what we're going to do is we're going to take this guy and we're going to go straight through another reaction. Now, I almost forgot something here. I forgot the 2 NAD pluses in my drawing here, producing 2 NADH. That's also how we're going to get our pyruvate. Then we're just going to regenerate that during this process. And I forgot to draw that in. Don't want to have to go back and try to re-edit that out. It's just easier to say, okay, well, since we did it right the first time, that's the mechanism. These 2 things happen to produce pyruvate. And then we're going to take this and produce 2 3-carbon compounds called lactic acid. So in here, there are no CO2s given off. So 1, 2, 3, 1, 2, 3. Now the thing with this is, since you're not giving off CO2, you could take lactic acid and convert it back to pyruvate in the liver in a process called the Cori cycle, and it can be used to shunt that back in. That pyruvate can then feed citric acid cycle, things like that, after it goes through decarboxylation of pyruvate, the intermediate reaction, all that. So it can go into a different shunt of metabolism. Now, who is it that does this? Well, mostly what you're looking at is a lot of bacteria like lactobacillus bacteria, but also animal cells. Animals, we do this as well. This is what builds up when you guys don't have enough oxygen and you do exercises without oxygen. So this is what builds up as that lactic acid. Now, or lactate. Now, what we want to do is, as you guys look at this, and I hope you take the time, if you sketch it out, it really helps you to sketch it in a simple form. And if you have this and you have to go into a biochemistry metabolism course at the upper division, then you'll be very well prepared for that. So that's kind of my goal, is to take these 2 processes here, sketch them out for you in an understandable, simplified way, so you have a foundation. Now, if you would like videos and more in detail on these biochemical processes, please let me know. I would love to draw them out for you, discuss them. But other than that, this concludes my video. Please don't forget to comment, like, and subscribe. Let me know what you would like to see in future videos, and thank you for watching.